Okay, so today I'm going to continue talking about aspects of antibody structure and function. You will see some things that relate to what we worked on in lab a teensy bit. Also remember that you have an inquisitive assignment due next week. It is posted and it's largely getting at aspects of antibody structure, a little bit on antibody function. So we left off last time thinking about aspects of antibody structure. I told you about the light chains and the heavy chains, each of which are identical on an individual antibody. We've got the two disulfide bonds holding together the two heavy chains, an additional disulfide bond holding each light chain to a heavy chain. The antigen binding sites are at the end that you can see up here. Sometimes we talk about that as the Fab, or the fragment with antigen binding. And we have this FC tail down here, which stands for the constant fragment. So just to kind of go into a little more detail about some of those structures. When we talk about the constant region, what we mean as far as constant is that there are not millions and millions and millions of types. Instead, there are 5 major types. Some of them do have some subtypes that I'm not going to really deal with a ton today, although they will show up on a couple of the slides. I really like to think about these as these 5 major isotypes. So these are 5 different types of heavy chain constant regions that we can have. One is M. It's encoded by a gene called mu, which is the letter M in Greek letters. We've got IgD, which is encoded by delta, IgG, which is encoded by gamma, IgE, which is encoded by epsilon, and IgA, which is encoded by alpha. And so you can see these 5 different types of heavy chains. We call them the 5 different isotypes of our antibodies. And the mnemonic that I use to remember them is MDs give everyone apples, because of that idea of an apple a day keeps the doctor away. So some other kind of important parts of thinking about antibody structure that we need to think about include thinking about the antigen binding section, but I do want to say one brief point about these regions that are drawn here, not with the rectangle of an immunoglobulin fold, but just kind of some protein. That is known as the hinge region. That hinge region is quite flexible, and that results in the antibody itself being quite flexible. So what you can see is that our antibody can actually flex in terms of how close together the Fab arms are. The Fab arms are able to rotate. The tail can sort of wag. And we can even see some bending of those Fab arms. Altogether, this means that the geometry of antigens can be accommodated by all of these different types of movements of the antibody. So the antibody is a bit flexible. It can flex as it needs to in order to bind to, say, 2 different examples of its epitope. But the part that I really want to spend a little bit of time talking about now is the variable region. And when I talk about the variable region, it is made up, as you can see, of both part of the heavy chain and part of the light chain. We do sometimes refer to this as the heavy chain variable region. this as the, oh sorry, this one's the heavy chain variable region. This is the light chain variable region. Or sometimes we'll talk about variable region in general. And when we say variable, this is the one where there are millions and billions and trillions of different versions, instead of the 5 that we saw with the constant region. The variable region is making up the end of the FAB, the fragment for antigen binding, and of course is thus responsible for antigen binding. In order to bind the antigen, the heavy and the light chain are coming together and combining to bind antigen. So the specific antigen that is bound by our antibody is making contact with both the heavy chain and the light chain. And which antigen this particular antibody will recognize is determined by the combination of heavy chain and light chain that is used. It's not just about which heavy chain, it's not just about which light chain. It has to be the combination of the two. So you can see that antigen is contacted at that antigen binding region by a combination of the heavy chain and the light chain. On old exams, you'll see that there are some places where I ask people to draw certain aspects of antibody structure, and I ask to draw an antigen interacting with the antibody, and every so often someone really sadly puts the antigen here. The antigen is binding to the antigen binding region that is made up of both the heavy chain and the light chain. You can also see that here as well. So you can see that there is basically some kind of three-dimensional shape that's made up from the heavy chain and light chain when they're put together. And that shape together is what will allow for being complementary to the antigen and antigen binding. These all kind of look like the heavy and light chain are nice and symmetrical. They don't have to be nice and symmetrical. But what I want you to see is that the heavy and light chain are coming together to bind to that antigen. What you can also notice here, as well as in other places, of course, is that we have some immunoglobulin folds. You can see these are shown as those same old immunoglobulin fold rectangles. Remember that immunoglobulin fold structurally looks like this, with multiple beta sheets, this disulfide bond holding it together, and some little connecting loops. And I mentioned that there were 3 little loops coming off the top. You can kind of think of it like 3 little fingers. So you can see I've got the 3 little loops up there. You can see the 3 little loops here as well. If we actually look more closely, this one. If we look more closely at how our antibody is binding to antigen, we find that in the end of those immunoglobulin domains, so the variable region of the heavy chain and the variable region of the light chain, there are those 3 little loops, like these 3 little fingers in each case. And it's actually those 6 loops together that are touching the antigen and that are the thing that are making contact with the antigen. So those loops are, in fact, the key things. The loops are shown here as these little red stripes. And they are coming together to touch the antigen. You can even see this here. Here you can see the 3 loops from the heavy chain. They've got an H there, so that's how I know they're from the heavy chain. And the 3 from the light chain. And here's my antigen, and you can see they're all coming together to touch that antigen and make contacts with that antigen. So those loops become incredibly important. I've called these regions of the antibody, which are the ones that are sort of in the darker color on my little stuffed antibody, called those the variable regions because there are many different versions of them. If we actually do some amino acid sequencing, we find out that there are parts of them that are more variable than other parts. But I'm going to answer Karamie's question first.
Where are the loops exactly on that diagram?
So these are actually the tips of the loops. These three are the tips of those loops that are touching the antigen, okay? So those three little circles are the tips of these three loops. If we actually do amino acid sequencing on all the variable regions in all the world, we find out that there are 3 parts of the variable region that vary the most. We refer to those 3 parts as the hypervariable regions because they're really, really, really variable. And you can that here as well. So we can take a variable region and we can do amino acid sequencing and look to see how much the amino acids vary from antibody to antibody. And you can see some places they vary some, and in some little spots they vary a ton. You can see those 3 spots where they vary a ton. The hypervariable regions, the most variable parts. If we actually look at the structure of the protein and try to figure out where are those amino acids in the structure of the protein, it turns out they're the 3 little loops. The hypervariable regions are those 3 little loops. They're the ones that are contacting the antigen, and that's where our antibodies really vary the most. And so you can see them kind of drawn here as well. Sometimes we refer to them, as you can see here, as the hypervariable regions. We can also refer to them sometimes as CDRs or the complementarity determining region, because they are the thing that determines which antigen our antibody is complementary to. We will see much more with those CDRs later. So I'm actually going to skip ahead. I'm going to skip over the next little set of slides. And I'll do them later if we have time. Later today if we have time. If not, I'll fit them somewhere else. I'm not going to worry about them right now because I would rather get into this part of the information. If you go out and you ask somebody about antibodies, you will hear lots of things that they say antibodies do. Sometimes I've had people explain to me that antibodies eat bad guys. And they kill the bad guys. And they do all sorts of great things to the bad guys. Right? They're like superheroes. If you ask people who are not scientists what antibodies do, you will hear these amazing things that these superheroes can do. There's a little bit of a problem, however. This is a chain of 4 proteins held together by disulfide bonds. It can't eat. How's it supposed to kill something? So now that we've actually looked at the structure, you can sort of look at it and be like, okay, but then how the heck does that little thing actually do anything about a microbe? And so that's what we're going to spend the rest of our time today talking about, are the ways that antibodies can actually do something about microbes. This image is showing 6 different functions of antibodies, 6 different ways that antibodies can get rid of microbes. And that works for me. Sometimes I think about 5, but some of my 5 overlap, so I'll go with 6. I'm good with 6. So that's what we're going to talk a little bit about right now. Happily for you, as we talk about the 6 functions of antibodies, you will realize that some of them already sound like things we've heard about this semester. So some of them are already going to seem familiar to you. So one of the functions of antibodies is that they can fix complement. So an antibody being bound to an antigen can start the classical complement cascade. So basically here the antibody is dealing with the microbe by binding to the microbe and sort of being a label that tells the complement cascade, start here, kill this. And so that's one of the ways that antibodies can have an effect against a microbe, is by starting the complement cascade, or an effector function. The second function of antibodies, again, is one that you're going to see at least be related to something we've talked about before, and that is opsonization. So when did we talk, or how did we talk about opsonization before? When I told you about opsonization, what was opsonization? We've talked about this term before. What? What were we going to say, Jay?
You gave an analogy of
There's an analogy. Yep, Rumi. So you mark the bacteria so the macrophages can eat it. My analogy was that it was like butter. It's putting a coating on something to make it more tasty and more likely to get eaten. And we saw opsonization before in that complement could opsonize things. So if you put a coating of complement on something, that makes that thing more likely to get eaten. It turns out that antibodies also can opsonize. So if you put a coating of antibodies on a bacteria, that also makes it more likely to get eaten. So it's the same idea of we're putting some kind of coating on the microbe and making it more likely to get eaten. Here the coating is an antibody, so we've opsonized with an antibody before we opsonized with complement. So again, we've got 6 total functions of antibodies. 2 of them are things you've seen before. Opsonization, in the case of complement, required that a cell have a complement receptor. So the macrophage had to have a complement receptor so that it could see that complement coating to phagocytose the cell. In the case of the antibody opsonizing, the complement receptor is not really going to work here. Instead, our cell has a receptor called an Fc receptor. And the Fc receptor binds to the Fc portion of the antibody. There are different Fc receptors. So there's, for example, Fc alpha receptors. It binds IgA. There's an Fc gamma. It binds IgG. There's an Fc epsilon. It binds IgE. So the letters are all matching up. And so an Fc receptor is required for opsonization to work. The third function of antibodies that I want to tell you about is the one that I will admit I spend the most time thinking about. It's the one that virologists care a lot about because it's really important for antiviral responses. So to me, this is like the really important one. And it is a function called neutralization. On the left, you can see a general example of a virus infecting a cell. So here you can see the virus interacting with some protein on the surface of the cell. that allowing the virus into the cell so the virus can start reproducing. If there are antibodies around that can perform this function of neutralization, those antibodies will bind to that virus, that extracellular virus, or extracellular whatever, and physically block that virus, or whatever, from interacting with its receptor and getting into the cell. So basically the idea is that those antibodies are physically getting in the way of this interaction between the virus and its receptor. Sometimes when I think about neutralization, I think about it as being really fancy steric hindrance. You can see another example of this here. This is a virus. This is rhinovirus, one of the causes of the common cold. It's binding to this cell receptor. You can see we're even color-coded with different proteins. And here you can see antibodies in green binding to that rhinovirus particle and making it so that this interaction is no longer possible. It can happen if the antibody binds to that exact site. It can also happen if the antibody just binds kind of nearby and is bulky and just gets in the way. But like I said, either way, it's basically just fancy steric hindrance for neutralization. When, say, people were testing COVID vaccines, they would measure first if you had antibodies, but then the second test they would do is, do the antibodies you make neutralize? They would look, do you make neutralizing antibodies? Because neutralizing antibodies are so important for antiviral responses. So antibodies that can do this function are great. As I said, we often think about them as virologists, but neutralization can work in other ways as well. Some bacteria cause disease by making different types of bacterial toxins that hurt our cells. So here is a toxin binding to a receptor. The toxin ends up in the cell and poisons the cell, as the figure says. If there was an antibody around that bound to that toxin, that antibody could neutralize the toxin. Keep the toxin from interacting with the receptor, keep the cell safe. So it's the same process of neutralization. You can see that it doesn't have to be something that has to happen with viruses. I told you about the von Behring and Kitasato experiment and how they were really lucky that they happened to pick bacteria that gave them a very clean answer. The reason why that happened is all three bacteria they picked work by toxins. And in all three cases, the antibodies they were looking at neutralized the toxins, which we know now. They didn't know at the time. So we've got 3 functions of antibodies so far. Complement fixation, opsonization, and neutralization. The next one is one that people often don't like. It's one that you hear about and you say, boy, that seems really lame. And I will admit there are some reasons why this one is not as useful as, or is hard to understand how useful it is. But what you want to remember for this one is that our antibody has 2 arms that can bind to the same epitope. And so if we have an antigen with, say, and our antigens often have multiple epitopes on them. So we can sometimes get situations like this where we have one antigen being bound by multiple antibodies because there are multiple epitopes on this antigen. And so you can see more than one antibody can bind. And each antibody could bind to more than one antigen with the 2 different arms. So you can see we could get these big clusters that can form. Those are known as antigen-antibody complexes. And those antigen-antibody complexes will precipitate out of blood or other body fluids. So basically what is happening here is the antibody is precipitating the antigen out of the body fluid. So it can't, say, flow through your blood and go somewhere. We've held it up in one of these big clumps. We basically made giant clumps of antigen so that antigen can no longer flow in fluid. There are also some downsides to making big clumps. If we filled your vessels with big clumps of antigen, that would not be great for blood flow. So there are some reasons why this is bad sometimes, but the idea here is that we're getting antigen-antibody complexes and basically precipitating the antigen out of solution. In many cases, those large complexes can actually be cleared either by complement or by phagocytosis pretty easily. So they usually don't end up just sitting around as clumps. But the thing that we're thinking about as the function of the antibody is clumping together the antigen to sort of get it out of solution. All right, so we got 4 functions of antibodies. There are 2 more. The 2 that I'm going to tell you about last have something in common. And they have something in common with opsonization as well. So all of those, so opsonization as well as these other 2 functions all require an Fc receptor. So they all have some kind of cell doing an action because that cell has an Fc receptor and can recognize there's an antibody tag here. So I should do something. In the case of opsonization, that thing was phagocytosis. And the cell was a macrophage or other phagocytic cell. We can also see other types of cells with Fc receptors get activated. Here we're looking at the activation of a type of cell called an NK cell, or a natural killer cell. The official name for this function is ADCC, or antibody-dependent cellular cytotoxicity. So this is a way of activating NK cells using antibodies. You can see that the NK cell has Fc receptors on its surface, which are shown in green. And those Fc receptors can bind to antibodies that are on the surface of cells. And so here you can see antibodies bound to the surface of a cell. Now, an NK cell with its Fc receptor can recognize those bound antibodies. And the NK cell will do its function, which is implied by its name, the natural killer cell, which is that it will kill this cell. It will induce apoptosis in this cell. And so this cell that was labeled by antibodies will get killed by our NK cell. You can see this happening here as well. For reasons that I don't fully understand, in the textbook they always like to show this as the NK cell killing a tumor cell, which in fact, yes, it can kill a tumor cell. That becomes this extra layer of complication that I feel like they don't need to tell you right now. I usually think of NK cells killing cells that are infected with viruses. But yes, it can be a tumor, as this is showing you as well. So again, the idea here is we've got a cell. It's got an Fc receptor. When the Fc receptor binds antibody on the surface of another cell, then the cell does stuff. The 6th function of antibodies is really similar, except now we don't have an NK cell with an Fc receptor. Instead, we have another cell called a mast cell. And the mast cell has Fc receptors. When those, and what actually happens with the mast cell is a little bit unique in that when mast cells are hanging out around your body, they have lots of Fc receptors. And if they happen to come across the antibody that matches to those Fc receptors, they end up with antibodies on them before there's any antigen, before there's any problem. So your mast cells right now are chilling with some antibodies on their surface, if they happen to find any antibodies that work for them. If then the thing that binds to those antibodies is present, that will send a signal to the mast cell. You can see that it's kind of bringing two of these receptors close together, making the mast cell signal. And the mast cell, which is full of all these granules, will just release its granules. It will secrete everything it's got, with the idea that it is generally trying to kill some large extracellular thing, like a worm or some other parasite. So you can see the mast cell is basically full of these granules, almost like little toxic bombs. It gets covered with antibodies on its Fc receptor, and if the thing that binds those antibodies is ever around, then the mast cell just releases all of its toxic bombs, all of its granules, to try to kill whatever is around. If you're like me, then unfortunately your mast cells have decided that the things that they want to try to kill are tree pollen and sometimes just your lungs in general and all the things you're allergic to instead of parasitic worms. All of the functions that we have seen of our antibodies thus far are largely dependent on the Fc portion of the antibody. The FAB is binding the antigen, but then we do something about that antigen using the Fc portion. So what we also need to talk about when we think about antibody effector functions are the 5 different isotypes, because it turns out that each of these 5 different types of antibodies has some particular things that are unique about them. And in a lot of ways, they are good, really, really good at one of the functions. And so we're going to try to now take the different isotypes and the different functions and sort of put them together. I have to tell you one other brief thing about antibody isotypes. Those of you who know much biochemistry are going to despise this next slide. The reason why you will despise this next slide is because you'll say, that doesn't make any biochemical sense. It does. It's just that I'm not going to explain how it works until later in the semester, because it's sort of like a problem I do not want us to deal with right now. We got other complicated things to deal with. We don't need this one yet. So basically what I'm telling you is that this happens. And you're going to be like, what? That shouldn't happen. You're right. It has a whole special thing, and I'll tell you about it later. Now, all I need you to know is it happens. So our antibodies are able to actually switch isotypes. So we can take an antibody, and we can keep the same antigen binding region, and we can actually switch which constant region that antigen binding region is paired with to get a different isotype. So perhaps we might have an antibody that is this awesome antibody that is IgM. And maybe over time, maybe we realize that that antibody binds super well to a parasite antigen, to some parasitic worm. And IgM is not the thing that's the best for parasitic worms. IgE is the best for parasitic worms. We can actually switch the isotype from IgM to IgE. Keep the same antibody binding area, but switch the constant region. We also can, in fact, improve our binding of the antibody over time. So antibodies get better and tighter binding over time. So their binding strength does change. Again, how that works will be a problem for later. Now, just know that it happens. We can class switch. We can switch different isotypes. We can affinity mature and make our antibodies bind tighter to a particular antigen. All right, so your textbook has 2 tables in it that talk about the unique aspects of different isotypes. First thing to note here is that this table, as well as the next table from your book, does divide up IgG into IgG subclasses. So you can see there's IgG1, IgG2, IgG3, IgG4. I'm not really going to focus on the differences between those. I'm going to say like one brief thing about the differences, but I'm basically just going to call them IgG. But in reality, there are multiple types of IgG. In fact, there are actually more types than this. You can see your textbook also does divide up IgA into IgA1 and IgA2. Again, I'm mostly just going to call it IgA. But to be correct, your textbook does tell you about the 2. What you can see in this table already is that there are some places where we see some big differences between these different isotypes. So you'll notice that the molecular mass is not the same for all of these isotypes. You can see that the serum level, as in how much of that isotype is in the blood, can really differ for our different isotypes. You can see that the half-life, how long that the antibody lasts in the blood, differs a lot for different isotypes. And you can see that the specific functions that these isotypes are good at will vary a bunch among the different isotypes. So we're going to now go through each of the 5 isotypes and kind of talk about what's key about that. I did pick them. They are not in the MD's Give Everyone Apples order. I put them in another order that I think makes more sense in this case. So hopefully that won't confuse you. But we are going to start with IgM. And as you'll learn later in the semester, your immune response always starts with IgM too. So kind of, I don't think I would know what to do with myself of not starting with IgM. If you look at this table and IgM, where do you notice right away that IgM is very different than the other isotypes? Yep, Christina. Yeah, the molecular mass of IgM is way larger than the others. So you can see that it's a big standout. It's kind of in the middle for some of the other things that are shown here. But mass, you can see, is a big difference. The reason that IgM has that change in mass is because IgM is generally not found as a single antibody. There's another protein called the J chain, or the joining chain, that holds together multiple IgMs. So IgM doesn't hang out as a single antibody. IgM hangs out as a pentamer. So we've got 10 different FABs and 5 different FCs all held together with this J chain. You can see that in cartoon form from your textbook, and your textbook in chapter 4 also shows you 2 different electron micrographs where you can actually see pentamers of IgM. And so this is why IgM's molecular mass is so big, is because it's always found in a pentamer. One of the things that's really nice here is that this means instead of having 2 binding sites for antigen, IgM really has 10 binding sites for antigen. And so that helps it have pretty strong interaction. We get a lot of avidity because of having those 10 binding sites. IgM is a standout in one other way as well, but it's honestly related to being a pentamer. When we think about the functions of IgM, IgM is really the all-star in activating complement. When I was putting this slide together, I was feeling mad at your textbook because they gave it 3 pluses here, and they gave one of the IgGs 3 pluses too, and I'm like, that IgG is not as good. Like if you're given IgG 3 pluses, this needs 4 pluses, or 5, or maybe like 10. Because IgM is so good at fixing complement. So much better than any of the others at fixing complement. The reason why IgM is so good at fixing complement is because it is found as a pentamer. So it can bind to multiple copies of its epitope on the surface of some microbe, and it makes this nice shape with the Fc portions that is perfect for C1q, or for C1. IgM has exactly the right geometry when it's a pentamer in order to bind to C1. So yeah, other antibodies that aren't a pentamer can kind of luck out and get the right geometry. But IgM, because it's a pentamer, is like the perfect binding place for C1. And so IgM is an absolute all-star at fixing complement. I'm going to move now to IgE. And if we look at IgE, you can also see a place where it stands out as being really different from the others on this table. So how does IgE stand out from the others on this table? Yep, Carney. It has a lower serum level. It has a really, really, really low serum level. The reason why IgE has such a low serum level is because it's found somewhere other than serum. It doesn't hang out free in the blood. It hangs out somewhere else. So this actually doesn't mean that there's no IgE in the body. This is just telling us it does not spend its time in the serum. It's, like I said, it spends its time somewhere else. And you can see, again, I don't like this either. I just like this one. You can see a unique thing about IgE here. So what do you see that's unique about IgE in terms of this table of functions? Yeah, Christina. It's only really good at one thing, and what's the one thing? Sensitization of mast cells. Remember how I told you about how the mast cells have this Fc receptor and soak up a whole bunch of any antibody that works for them? Yeah, they specifically have an Fc receptor for Fc epsilon, and they specifically soak up all your IgE. So your IgE doesn't hang out in the serum. All of your IgE right now is coating your mast cells. That's why it has this low serum level. The mast cells don't have Fc receptors for any other isotype. So the only isotype that has the ability to turn on mast cells and do this reaction as shown here is IgE. So IgE is our all-star at sensitizing mast cells. And so again, as I am allergic to everything, I want to have a conversation with my IgE and know why it is doing this to me. So I mention IgE and mast cells, partially to help me out with the next isotype that I'm going to talk about. With the next isotype that I'm going to talk about, I'm going to show you a table just like the table I've been showing you, where I put a red box around the isotype and you see the information. But I am first going to show you that table from an older textbook. Okay? So it's going to look different because it's a version of a table from the older textbook. There's a reason. But know that I'm doing this on purpose. And so this is kind of the old version of IgD. So if you look, oh, this one, it's even slightly updated. Man, this is not old enough. Oh. If you look at the oldest IgD table that I have that I apparently could not copy in here correctly. When I was a student learning about IgD, IgD had a minus for everything. And IgD was the one that I learned as has no function, or has no known function. We often talk about IgD as potentially an evolutionary relic that was useful in the past evolutionarily and that we still have, but doesn't necessarily do a whole lot. And most immunologists, when they think of IgD, they think about it as being a flow cytometry marker that we use to name certain B cell subsets. And like it actually doesn't do anything. It just so happens that it is on certain kinds of B cells, and we use it for B cell marking. And so you can see different B cells have different stuff on their surface, and special ones have IgD. And so honestly, when I learned all of this, and until 2005 at least, later than 2005, 2010 at least, my answer was IgD has no function. It just happens to be a thing we use in flow cytometry sometimes. People have done some additional studies of IgD, and they have found one function for IgD. I have no textbook pictures of this function or anything like that. I just have to tell you about this one function. So the one function of IgD that we now think about is shown here, and it is the activation of basophils. And we think about that activation of basophils as being pretty much the same kind of thing as IgE is doing with mast cells. So that's why I told you about IgE with mast cells a little bit ago, because we think IgD is doing the same thing with basophils. This was only discovered, like I said, maybe in like 2008 or something. While the IgE mast cell thing was discovered in, I don't know, like 1900. I made that up, but it was a long time ago. Why did it take so long to come up with this function of IgD? Yep, Jameer. Basophils are super rare. Remember when we looked at cells in the blood and pretty much nobody found a basophil? Well, if you can't find a cell, you can't study how it gets sensitized by IgD. And so as far as we could tell, IgD didn't do anything. So there's a lot we don't really understand about what IgD is doing with basophils because they're just so hard to find and so hard to work with experimentally. But this does seem to be the function of IgD. All right, so we've got 3 isotypes down, 2 to go. The next one that we're going to talk about is IgA. So on this figure, I think that if you looked at IgA, I would call IgA pretty average here. I don't think that IgA seems to stand out a whole heck of a lot on this figure. But IgA does have some things about it that are really, really important. you will notice that it is good at neutralizing, though it's not the only one that's good at neutralizing. So it's a good neutralizer, yay. But the thing that's really interesting about IgA is where we find IgA in the body, IgA's anatomic location. So if we think about our antibodies, this is going to be, I mean, I am known for bad drawing, but this might be like over the top bad drawing. I'm trying to think about how I even try to do this drawing. So if we zoom in on a person, we'll see the blood vessel, and we'll see sort of some tissue, right? And maybe we'll see, maybe this will be like the GI tract. This will be the blood. This will be some other tissue, right? So we're zooming in on this person. The cells that are making the antibodies are making them in the blood. And so we've got antibodies and antibody-producing cells hanging out in the blood. And there's a wall between the blood and the other places of the body. IgA can get across some of those walls. And so IgA can be found in locations other than the blood, because it can get across some of these barriers, these epithelial barriers. IgA is also generally thought of as a dimer. It also binds to the J or joining chain, and so we get dimeric IgA, where we can get 2 molecules held together. So we've got 4 binding sites. And like I said, IgA's big thing that it can do is it can get transported across cell layers to end up in other body sites. And specifically, those body sites are mucosal body sites. So IgA is found in your lungs. IgA is found in your nose. IgA is found in your saliva. IgA is found in your genital tract and urinary tract. You can see IgA here. So while most of our other antibodies kind of hang out all over the body in the blood, IgA is unique in that it is in all of your mucosal sites. So it's kind of helping those anatomic locations have that first line of defense. It almost helps them be barriers because it's there while none of the other antibodies are there. Also, interestingly about IgA, IgA is found in breast milk. And so mothers who breastfeed are also giving IgA to their babies so that their babies do have IgA in their GI tract. When IgA is in these anatomic locations, its function is to neutralize. So if you didn't have IgA, you might have microbes growing in your mucosal area. I believe this is supposed to be a pharynx, but it doesn't really matter to us. But because we do have some IgA in those mucosal areas, we can actually neutralize bacteria before they're even able to get into the rest of the body and replicate. So we can, in fact, block infection in a lot of ways with this mucosal IgA instead of waiting for the microbe to get into the blood where the rest of the antibodies are found. So it's kind of a nice early system. It's actually part of what makes the barrier organs barriers. And may be important for preventing disease. One of the big issues that people talk about with, say, your COVID vaccine is that it can prevent you from getting sick, but it might not prevent you from getting the virus. Like you might get the virus but not get sick. The reason is because it doesn't always give you good IgA in your nose. So there's no IgA in your nose. The virus can get in and go for a couple cells, gets to the blood, and the IgG, or the antibodies in the blood, are like, ha, gotcha. But it can't actually stop anything from here. If we had a vaccine that could give you IgA, which some people are experimentally working on, then perhaps it would even, that you would already have antibodies, say, in your saliva, in the mucus of your nose, that could stop the virus from even getting in the first place into the first set of cells. So IgA and how we make your body make IgA versus other isotypes is an important topic for many immunologists. One other reason why IgA is really interesting is that there is something called selective IgA deficiency, where people do not have the ability to make IgA. Selective IgA deficiency is the most common immunodeficiency. You can see numbers of individuals with selective IgA deficiency from, this is per million of individuals with different types of backgrounds here. And so you can see in some cases it's somewhat frequent. This is by far the most common. As far as we know, individuals with selective IgA deficiency, so who are completely missing this isotype, are totally healthy. You could be sitting here right now with no IgA and not know it. We think the reason for this is because of modern hygiene. There is some kind of mathematical association with maybe lung disease or celiac disease in people with this, but it's actually not super clear. For the most part, these people seem completely healthy. And one thing that we talk about, or we wonder about, is, okay, it's super weird that this is so common. There's a gene mutation giving people this deficiency, and this gene mutation is way more common than it should be. You know, we sort of might say, why is it that natural selection hasn't removed this mutation? And what we can realize is that, well, the time when you're really, really likely to have a problem with microbes, the time when you're most vulnerable is when you're a baby, because you haven't had the chance to make a great immune response to a lot of things. When you're a baby, it doesn't matter if you can make IgA, especially like historically, like with prehistoric or primordial, or I don't even know what the correct word is right now, humans, ancient humans. Because all babies got breastfed. And so babies didn't have to make their own IgA. They were all getting it from their mom. And so we think that the reason why this mutation was not eliminated from the human population is because it actually can't affect you until after you've finished breastfeeding. And the selective pressure may not be high enough at that point because you have other parts of your immune system playing a role. But again, it's not really well understood. We don't know why this is so common or what's going on with it, but it is one that, like I said, you could be sitting here right now and not know that you don't make IgA. Yes?
So basically, IgA deficiency is a gene mutation?
So there is a mutation in a gene that leads to you not having IgA, yes.
Okay, and how would you test for if you do have IgA?
I mean, you could take some, you could take blood samples and then like look for each of the different isotypes and see if one was missing. Yep?
Is IgA in baby formula?
It is not in baby formula. So you can imagine that's one of the kind of pieces that people talk about with breastfeeding versus formula. There is no IgA in formula. So the final isotype, which I will have a chance to start telling you about, but I will clearly not get all the way through, is IgG. one of the things to notice about IgG is that we've got a lot of subtypes of it. And if you looked at the serum level, most of the subtypes are on the high side in terms of serum level. So if you actually added that together for all the IgGs, you could see IgG is the one you have the most of by far, right? Because you have 12, 13 and a half compared to 1.5, 0.03. So IgG is really the main antibody in the blood. In fact, most people, when they're measuring antibodies, looking for antibodies, thinking about antibodies clinically, they're thinking about IgG. IgG also sticks around for the longest period of time. It has a very long half-life. It's pretty good at a number of different functions. So it's a great neutralizer. Some of it can opsonize. Some of it can turn on NK cells. Some of it can turn on complement, all that good stuff. The subtypes largely vary based on the hinge region, how long it is and how flexible it is. That also does things like change how easy it is to degrade. It's really easy to degrade IgG3 because look at all those places a protease could cut, while some of these others are much harder to degrade. So those are the types of things that vary between the different subtypes. When I think of IgG, one of the big things that I think about is that IgG is made a little bit later in a primary adaptive response. But it is the main thing that is being made in a secondary response. So if I want to measure if you are making kind of a good response after vaccination, I'm going to look and see if you made IgG. And part of that is just because there's a ton of IgG, and so it's easy to measure. And part of it is because it does get made a little bit later in the response. I will say a couple of things to start out with about IgG as well, because it does have one other unique feature, and I'll start with that on Monday, but I have one really important thing otherwise to tell you about Monday. On Monday, I'm going to say a few things about IgG, but then I'm going to start talking about something known as the antibody diversity problem. We'll be talking about the antibody diversity problem Monday and Wednesday. For some of you, the antibody diversity problem will be the hardest thing we do this semester. There are some people who it really breaks their brain a little bit. I highly recommend, if you are not a person who reads before class, that in this case you do the readings before class for Monday and Wednesday, so that you have heard some of it before, and as I'm up here talking, you're like, what is this foreign language she's saying? I want you to have at least heard it once before. So definitely be ready for Monday and Wednesday as we talk about diversity, and I will see you guys then. Remember, the inquisitive is coming up for next week, but also please read and kind of start thinking about the diversity stuff before class on Monday. I will see you then. Hope you have a great weekend.